Hello, my name is Deborah and I'm going to be giving a presentation on respiration. Now, when we say respiration, most people think of it as the breathing in of oxygen and the breathing out of carbon dioxide. Others might think of it as the breaking down of food molecules into energy. Both are right. And my biology textbook puts it in a much more scientific way by saying it's the enzymatic release of energy from organic compounds in a living cell. Now, before we delve deeper into respiration, I'd like to talk about a part of the cell that plays a huge role in respiration. It's called the mitochondria. Now, it's made up of the outer membrane, which is smooth and is relatively permeable to small molecules. We then have the inner membrane, which is less permeable to smaller molecules and is folded inwards to form cristae. This is to increase the surface area of the inner membrane. Then we now have the space, the intermembrane space between the outer membrane and the inner membrane, and it has a lower pH than the matrix, which is the matrix is the space inside of the inner membrane. Now back to respiration, now that we know what the mitochondria is. Respiration was defined as the enzymatic release of energy, organic compounds in living cells. So we normally call energy ATP. So ATP is made up of adenine, which is a nitrogenous base. It's made up of ribose, which is a sugar, and 3 phosphate groups. Respiration goes through many steps in order for us to get the final product of carbon dioxide, water, and energy. First of all, the first process it goes through is glycolysis. Now, glycolysis is the lysis of glucose. Lysis means to break or split. Now, glucose is made of 6 carbons. 1, 2, 3, 4, 5, 6. Now, we need to try and break this into 2, but it's tightly packed together because glucose is fairly stable. So we use ATP. When we add 1 ATP to it, it loosens the bonds a bit, and it now becomes fructose phosphate. Now, when we need to add a second ATP because it doesn't seem loose enough to break apart. So when we add the second ATP, it now becomes fructose bisphosphate and the lysis occurs. Now we have 2 3-carbon molecules, which we call pyruvate. Now this pyruvate, each pyruvate releases 2 ATP and 1 NADH. And this process occurs in the cytoplasm, which is outside the mitochondria in the cell. Now these 2 molecules of pyruvate go into the link reaction. Now the link reaction takes place in the mitochondrial matrix, which was inside the inner membrane of the mitochondria. And now our aim is to reduce these 3 carbons to 2. So what do? We decarboxygenate it, which is the removal of carbon dioxide from it. Then we also dehydrogenate it, which is the removal of hydrogen from it. Then now we add coenzyme A to it. So we now have a acetyl coenzyme A, which has 2 carbons, which then now moves on to the next step, Krebs cycle. Now it has 2 carbons. It's now 4 carbons is now added to it to form citric acid. Since it's a cycle, it's going to have to lose 2 carbons in order for it to have 4 to redo the cycle again. So that's what happens. It's decarboxygenated twice, which is the removal of carbon dioxide. It's dehydrogenated 4 times, 3 with the carrier NAD and 1 with the carrier FAD, and it releases 1 ATP. This is all per cycle, which is for per molecule of coenzyme, acetyl coenzyme A. So now for 2 acetyl coenzyme A's, we have the production of 6 NADHs, 2 FADs, and 2 ATPs. And the Krebs cycle happens also in the mitochondrial matrix as the link reaction. Now, all of the hydrogen carried by the carrier molecules now move on to the next stage we call the oxidative phosphorylation. Now, this takes place in the inner mitochondrial membrane. Now what happens is these hydrogen that are carried by these molecules are separated into the electron and the proton. Now the electrons pass the electron transfer chain and go in and oxidized to form water. Now the protons move into the intermembrane space, because all of them are probably like feeling congested in the matrix and want to like spend time alone. So they move one. Now this hydrogen proton is thinking it's going to be alone, only to be followed minutes or seconds even later by another hydrogen molecule. And then by the time they realize that hydrogen proton is not getting the amount of space it thought it would get, so now it wants to go back into the matrix, but it's not able to pass the proton complex that it passed when going to the intermembrane space. So it has to pass another place called the ATP synthase. Now, as each of these hydrogen protons are passing through this, ATP is formed. How is it formed? Well, there's this turbine-like structure in the ATP synthase. So as each hydrogen molecule comes, it turns, ATP is formed. Another one comes, it turns, and ATP is formed. The ATP is phosphorylated with the energy from it. Now this step alone produces 28 ATPs. Now what if there is no oxygen? Then we have something we call the anaerobic respiration, which occurs when there is no oxygen. Now this produces way less ATP than aerobic respiration, as most of the processes of respiration need oxygen. And and when oxygen is not there, almost all of it is cut off except for glycolysis. So now what happens in anaerobic respiration is either alcohol fermentation or lactate fermentation. Now in alcohol fermentation is common in microorganisms like yeast. So what they do is they convert the pyruvate from glycolysis into ethanol, while in mammal cells, this pyruvate is converted into lactate acid. Now the ethanol in yeast cannot be reversed, but the lactate acid in mammal cells can be reversed. Mammal cells don't produce ethanol when there's no oxygen, because if that were the case, then every time we were to work out or do some strenuous exercise, then we'd get drunk, because our body is converting that pyruvate into alcohol instead of lactate acid. But rather our cells convert it into lactate acid, which is converted back later on after your workout when you start to take in more oxygen and oxygen is present. We move on to respiratory substrate is a molecule from which energy can be liberated to produce ATP in living cells. Now some cells use lipids or amino acids instead of glucose. So most of the energy that is released in respiration comes from the oxidation of the hydrogen molecules to water. So now that should tell us that if a respiratory molecule has more hydrogen per molecule than another, it's going to produce more energy than the other. For example, fatty acids and carbohydrates. Fatty acids have more hydrogen, so will have a higher energy density than carbohydrates. We can calculate the energy of a substrate by burning a known mass of it in oxygen in a calorimeter. Now we have the respiratory quotient, which is carbon dioxide on oxygen. Now this shows us what substrate is being used, and it can also tell us whether aerobic respiration is occurring or anaerobic respiration is occurring. How does it tell us this? When anaerobic respiration is occurring, oxygen is going to be 0. That means we're going to have a number for carbon dioxide, say 2, all on 0. And that's going to give us infinity or undefined. So that should tell us that it's an anaerobic respiration. Meanwhile, if we were to have aerobic, we'd get an answer because it might be maybe 2 on 1. So we'd get an answer. Here's a short summary of all that we learned. Respiratory is the enzymatic release of energy from organic compounds in living cells. The mitochondria is made up of the outer membrane, the inner membrane, the intermembrane space, and the matrix. ATP is made up of the adenine, ribose, and 3 phosphate groups. Organic compounds are broken down through glycolysis, which occurs in the cytoplasm, the link reaction, which occurs in the mitochondrial matrix, the Krebs cycle, which occurs in the mitochondrial matrix, and the oxidative phosphorylation, which occurs in the intermembrane space. Now, if oxygen is not present, then alcohol fermentation or lactate fermentation occurs. So that brings us to the end of my presentation. Thank you guys so much for listening. Bye.